Hello and welcome everyone to this epigenetics podcast. In this podcast, we're going to talk about 4 different mechanisms to explain how DNA methylation affects transcription. In the last podcast, I talked about the general process of how methyl groups were added to cytosines and how certain regions were more or less likely to be methylated and what that meant to the overall transcription. But I didn't talk about how. And there's lots of different mechanisms that have been proposed, and it's probably a combination of many of them. Okay, of the 3 mechanisms that we're going to talk about, the first one I want to discuss is how methylation can affect chromatin structure. Observations have been made that unmethylated DNA, I'm just going to draw it like this, unmethylated, has an open and accessible conformation, meaning transcription factors have access to this DNA. Other enzymes that need to bind here to affect transcription have access to this DNA. It has a much more open and accessible structure. Whereas DNA that is methylated is proposed to be, so let me write methylated here, and I'll draw some M's here for methyl. It tends to be a little bit more closed. It tends to have more histones associated with it. It is inaccessible to various transcription factors that would like to bind here. So it's possible methylation affects the overall chromatin structure, and in doing so affects whether or not transcription factors will be able to bind to appropriate regions on the DNA to turn genes on or off. Okay, this second mechanism is a little similar to the last one. In the last one we said that adding methyl groups to the DNA changed the structure of the chromatin so that transcription factors could not bind. This mechanism states that DNA binding regions are blocked by methyl groups. In the last podcast, I introduced you to these terms, the CpG repeats, cytosine phosphate group guanine, cytosine phosphate guanine, so forth. It could be that transcription factors recognize this sequence, or a sequence similar to this, but certainly a sequence with a lot of these CpG repeats. This is the sequence these transcription factors recognize in order to turn on the transcription of this gene down here. So this is a sequence of DNA here. CpG repeats, transcription factors recognize these. They recruit in other transcription factors, RNA polymerase, to turn this gene on. This mechanism would state that the binding of methyl groups to these cytosines makes this region here that this transcription factor recognizes sort of invisible to it. It no longer recognizes this region because these cytosines have changed, and then as a result we cannot turn this gene on. So the first mechanism says there's a chromatin structural change. In this mechanism it's saying that the CpG repeats are necessary for some transcription factors to bind. Some evidence that would support this is that there are several transcription factors that are known to recognize promoters or promoter regions with a lot of CpG repeats in them. These include AP2, c-Myc, E2F, and NF-kappa B. All of these transcription factors recognize promoters that have several CpG repeats in them. Interestingly, all of these transcription factors have a link to specific cancers, which ultimately will bring us back to this later in the semester of talking about how methylation and epigenetics in general is linked to cancer. Now, just to make life a little bit more complicated, even though we have these transcription factors that we know recognize promoter regions with these CpG repeats, there are other transcription factors, like one called SP1, that doesn't recognize CpG. This just shows that not all transcription factors are going to be activated in the same way. Okay, let's talk about this third and final mechanism. And this mechanism suggests that DNA methylation attracts large repressors. These would be large repressing factors. These large aggregates of proteins that come together to have a common function, like we talked about before with the SNF factors. And what this mechanism suggests is that we have our DNA here, a 5' 3' end, and that within these CpG repeats, it contains these methyl groups on the cytosines, and these methylated cytosines act sort of as a target for large repressors. They wouldn't recognize a region down here, CG, that's not methylated. They require the methyl group on the cytosine. To give a little support for this mechanism, there are two repressors, MECP1 and MECP2, that are repressors like this that specifically recognize regions of DNA that contain methylated cytosines. And they don't bind to regions, similar regions, that have the cytosines unmethylated. Okay, so I want to say a couple things here about MECP1. The first is this term called threshold. Whether or not MECP1 will bind to these regulatory regions of genes is dependent upon how many CGs are here. If it's just a few, MECP will not bind. But if it's many, and the book isn't clear and the literature is not clear what I mean by many, but for our purposes, I'll just say many CG repeats. It's a little vague, but that's what we'll work with. When it has many CG repeats here, MECP1 is able to bind here with greater strength and is able to exert its repression of these genes to a greater impact. Another thing I want to say about MECP1 is that if mutated, if MECP1 is mutated, then these genes, and when I say these genes, what I mean are genes that have a lot of these CG repeats in their promoter regions. So these genes are overexpressed, even though they're hypermethylated along here. Without this large repressor, the MECP1, the methyl groups have no effect. Now, as I said, this is a large protein factor. And so the next thing I want to say is that they have found a protein in this large factor, and I'll just draw it here, called PCM1. And what PCM1 does is that it is the protein, the specific protein in this large complex, and it binds methylated cytosines. That's its job. It's a pretty important job for this complex because that's the whole point. It's recognizing these regions here. Also in this complex are proteins that likely, we don't know for sure, but likely bind to the DNMTs. And we'll talk about these later on, but the DNA methyltransferases are the enzymes that add these methyl groups. So perhaps in this large complex, these DNMTs come along here and they help stabilize or form these methyl groups here so that the PCM protein can bind to it more effectively. Okay, now I'd like to spend just a little bit of time talking about the counterpart here, MECP2. It's a little different than MECP1, in fact perhaps quite different. One of the differences is that it's more abundant in a cell. There's more of this in a cell. Doesn't mean it's more important, it just means it's more abundant. This is probably the most striking difference, and that is MECP2 can bind single CpG units. And when it binds it, so binds CpG far from the promoter, suggesting it may have some role in disrupting that pre-initiation complex. Now I want to talk a little bit more how repressors, when bound to DNA, when bound to methylated DNA, how that can repress transcription. Okay, I'd like to close this section with a better explanation as to what these large repressor complexes are doing when they bind methylated cytosines. What are they doing? We know they can bind here. We know that when they bind here, they repress transcription. But how? And that how can at least partly be answered by understanding the subunits found within this large complex. Because remember, this isn't a single protein. This is many proteins coming together to accomplish that one task of repressing this gene. So I'm going to list a few examples of what we might see in here. PCM1, this is a methyl binding protein. I'm just going to put BP for short. So that's a protein that could be here. I'm just going to put it here for lack of a better place to put it. We also have enzymes known as histone deacetylases. We haven't talked about these yet, but we will. And one of their jobs is to maintain the histone's affinity for the DNA. So they help keep repressing. We also have some subunits that I'm just going to classify as known protein repressors. Meaning in other studies, they've known that, say, this protein here binds to this subunit, but they've always known that this protein is linked to repressing transcription of certain genes. And maybe this subunit is interchangeable with other repressor proteins so that this whole complex can be used at multiple genes. There might be chromatin structured proteins that help, when they bind here, maintain this ordered chromatin, which will maintain the repressed state. Another one I want to talk about is ATPase. It's not uncommon to have an ATPase as one of the subunits of this large complex to help provide energy that might be necessary to maintain this chromatin structure, to maintain the histone deacetylase's ability to maintain the histones on the nucleosome. So that's often a common protein. In looking at your podcast, I noticed I asked you to talk about this protein complex called NERD. And what the protein complex NERD is a histone deacetylase. So together, when we look at all the proteins that can form in this large repressor, because it's binding to these methylated cytosines, together they work to accomplish the goal of repression. Any one of these proteins by themselves wouldn't do it. But together in this large complex, because of genes like these MBPs, these methyl binding proteins, come along here and bring in the whole complex. Okay, that ends this podcast. In the next podcast, we're going to spend some time talking about the specific enzymes that add these methyl groups or remove these methyl groups. Okay, if you have any questions about what was talked about in this podcast, please make sure you come see me. If not, I'll talk to you later.